I'm going to start with a slide that essentially echoes what Ali Stiegel talked about in June at Stockholm, which is what's the basis of experimental design. So I come from a machine learning and statistics community, and when I hear about experimental design, I think of Bayesian optimization and online or active learning. And what this means is basically we have, in this case, we're going to have HCA. We're going to have an atlas, a set of experiments that describe the assays of single cells. And we're also going to have a toolbox of methods that we can use on these experiments. And then we're going to have a set of possible experiments in the future, okay? And we're going to put them all together in some online active learning method. Which is going to be a machine learning approach to put together all these ideas. And I'm going to talk about what that is. And then, as we've been talking about, we're going to feed back this information and be able to design a new set of experiments. And the point I want to make here is I think there's a real opportunity, given the heterogeneity of experiments, given the big open questions that this consortium is trying to ask about how these methods can be used in conjunction with the data that we have and the potential data that we could get to really help design and guide the way forward. So in the white paper that was presented yesterday, there's a phrase about how we're going to do this, and I think it also echoes these methods. So it says the experiments are going to be adaptively re-estimated, and I think that really hits at what these methods are trying to do, in that given the data that we have and given the experiments that we could do, we're going to continue to iteratively update the ideas for how we're going to do this moving forward. And so, again, I think this really is an opportunity for us to be the first consortium where we use formal statistical methods to think about experimental design. This really allows us to have a systematic and sort of uniform approach to solicit, evaluate project proposals, and think about integrative proposals. Think about how we can get all the groups that are working on this project working together toward a unified goal. I think the key point about this project, unlike the Human Genome Project where the goals were very clear, unlike even GTEx where I feel like the goals are fairly clear, where we know we want to optimize the number of cis-QTLs across tissues in the human body, I think there's not really a uniform objective here. I think there's a lot of different objectives, and we're going to go through some of those today. We've already heard a lot about them. But because of this homogeneous objective, we need to talk and consider it very clearly before moving forward with something. So what are the dimensions of variability that we've heard about in the last couple of days? Well, obviously, cell types and tissue types are a big one, and context plays a big role there. We need to think about where we're getting these samples from, so whether it's primary cells versus cell lines, iPSCs, organoids, all of these have been discussed in this conference so far. Single cell, single nucleotide, bulk sequencing have all been discussed. And in fact, my understanding from talking with a few of the speakers offline is that a lot of bulk sequencing is being done on these samples, but just not discussed. And if we can integrate those into the study, that might be interesting. Disease versus healthy, I should say that italics represents metrics that I think may be cell type specific, questions that are cell type specific, and brown represents ones that I felt from the white paper may be pushed to round 2. So disease versus healthy samples, fetal, pediatric, adult, geriatric donors, all of these are going to make a big difference. Post-mortem, transplant, and living donors. Experimental and platform technologies. This is obviously an incredibly crucial one, and I think we need to really consider the different goals and observations that we can get from each of the technologies that we have right now and the ones we're going to see in the future. Also non-human organisms, which we're going to talk about, it sounds like, in November. Other ideas that I think are going to be very system dependent have to do with genotype variation. Ancestry variation, as was talked about 2 days ago, evolutionary considerations, exposures and perturbations. This may be particularly informative in immune cells with a bunch of studies coming out on this already. Sex-specific tissue types, which it sounds like we're actually thinking about in the first phase. DNA and somatic mutations. And then bulk RNA, chromatin, protein, other types of observations that we can get from these cells. So all of these questions are ones that may play into our global objective. And again, we need to figure out how to trade them off and compare them to get the most sort of bang for the buck, if you will. There's another axis of understanding here too, which is this three-dimensional structure. So when we think about starting these, including imaging technologies, thinking about the different layers of resolution, I think we also have a long way to go to understand how these methods are going to fit together in a unified way to be able to inform the decisions and the goals we make in this HCA pilot project. So I want to use a very quick dictionary metaphor. So let's say I want to build a dictionary of words, not necessarily definitions, but just a set of words that we use in English language. So let's say we have one person's text, and I just pulled this from the internet. This isn't actually mine. The question is, if I'm going to build a dictionary, let's think about some of the design considerations we want to do. First of all, how many people's text should I examine, right? I know that I'm going to have a specific set of typos in mind. I'm not going to talk about particular topics. I'm going to talk about a lot of topics, or a specific set of topics a lot. When should I be confident in a word? How many times do I need to see that word in one person's text versus a series of people's text to be able to call it a word? Or when does it continue to be sort of a mistake, an error? How rare is rare? Right? I think this is a big question when we're thinking about the possibility of observations and errors in these technologies. When do I stop? This has been raised a bunch of times, and I think it's also critical. When do I say, yes, this word is officially a word, and I don't need to search anymore for it? And what data are too noisy? So I feel like with bulk RNA sequencing, we have a really nice idea about how to evaluate the quality of every single sample that we process, but in single cell is going to be a little bit more difficult. So while this is a very gross metaphor, I think it sort of hits on a lot of the ideas, a lot of the questions that we have for how we're going to put these data together and how we're going to be sort of statistically confident in our observations. So let me tell you just for a few seconds about what I think of when I think of an experimental design framework. So this specifically comes from a review paper from 2010 that's on arXiv. Please look it up if you're interested. But these are ideas from Bayesian optimization. And let me tell you just for a second how they work. So this is an iterative process. So here is the first iteration where we have two observations. So these points on this curve are observations. And the x-axis here is going to be one of the parameters for our experiment. We can naively think of it as the number of cells we're getting from a particular experiment. And the y-axis is going to represent the value of the objective function for that particular experiment. So say here we have 700 cells, and that has a certain value in terms of our objective. And our objective here is not well defined. Again, we need to think about what that is. But let's say it's some measure of number of unique sample or number of unique cell types and cost trade-off. So it's going to be some nonlinear function. So if we see this particular observation, and then we have two different experiments at two different number of cells, then we have two values of this objective function. Okay, so we're going to model this as some nonlinear function. We're also going to model the error. So in other words, we have a lot of uncertainty around this region because we haven't actually evaluated a particular sample at that many cells. So there's this next green line, which is something called an acquisition function. And an acquisition function, I think, is actually equally important to the objective function. What this says is, how much do I want to explore versus exploit? If I'm trying to get to the global maximum of this uncertain, very noisy, very nonlinear objective function, where's the best place to sample next, given that samples cost money? In other words, each experiment costs money and costs time. And I only have a few of these to do. So the acquisition function is going to trade off uncertainty. So it's very uncertain in the middle of this region right here whether there's going to be possibly the global optima is going to sit there. But we also know from this experiment right over here with very little uncertainty around it that already is a good place to sample from. So the acquisition function is going to, again, trade off this explore versus exploit question, whether we should explore the places that we're uncertain about for the global optimum or exploit what we know about already the experiments that we have. And the way it does that is by essentially pulling the error and the noise, the variance, and the current observations. And I should say, too, that as much as we're assuming that our observations have zero noise in them, I think in the objective function that we're talking about for this group here, we're actually going to have substantial noise. So you can see that this is an iterative process. We'll choose an experiment, we'll put that experiment down, refit this function, and then our acquisition function will look quite different. And now it's proposing that we explore over in this region to see if the global optima is there. And we keep on going in this iterative process, again, each time updating what our objective function looks like. So what is our objective in this project? And looking at the incredibly exciting and far-reaching goals of this project, I don't think it's as straightforward as some of the projects that all of us have been involved in before. And if we're really talking about going from genes to drugs, like I think is one of the objectives from the white paper, we need to think very broadly about exactly how we're going to trade off the objectives with the sort of cost of doing the project. I think equally important, like I said, is this idea of an acquisition function. So I'd like to stress here that let's let uncertainty and variability actually be our friend here and guide our experiments. So if we can really nicely characterize where in this objective function space we are uncertain, where we haven't sampled, it might be very exciting to dive right in there and try and do those experiments and see how they work and see how they're integrated in this objective function. Given, again, from the white paper that we're interested in reproducibility, integrity, and predictive power, I think these three things, again, play very nicely into this idea of letting uncertainty be our guide. And all of those are gonna be something that we can look at specifically from this objective and acquisition function as we define them. Another thing that I think that we have worked on in machine learning, we being the field, not me particularly, is this idea of a multi-scale dictionary. So we keep talking about the skydive, which I think is a beautiful concept. And I feel like, again, if we talk about multi-scale dictionaries, we can start out at this very, very high-level dictionary where we have a sort of set of words, a set of ideas, let's say. And then we drop down in the skydive to look at very, very particular topics and concepts that play an important role in defining and finding maxima to our objective function. I think other questions that, as a statistician, I'm really interested in looking at is, what does the tail look like? What does it mean to have all cell types? Where does abnormal cell type fit into this tail, and how deeply do we want to sample those? What do we do with incidental findings? So actually one of the most beautiful ideas I think that came for me out of the GTEx project with all of the incidental findings that we had, the many individuals that had undiagnosed cancer or specific interesting phenotypes in their tissue morphology that we were then able to study in this sort of incidental way. And I think, again, when do we stop? So how deep is deep enough? How broad is broad enough? How many individuals is good enough for the pilot phase? I think all of these are important questions. And again, I feel like we can use statistics and use uncertainty to guide the answers here. So one particular question that I think is going to prove incredibly powerful in this pilot phase is the idea of power calculations, especially to think about diversity. So when you think about a power calculation, we're basically talking about the probability of identifying a specific question at a certain depth. So in this case, if we have number of cell samples, I should say this is beautiful work from the, I'm going to pronounce your name wrong, Satija lab. This is an online method. So sorry, I screwed that up. Okay. Okay. So this is an online power calculation for how many cells you should sample for a particular experiment. And I just poked a number of parameters in here and tried it out. And I highly recommend you try this out as well. This is a beautiful first pass. And I would like to integrate ideas to this in some kind of experimental design approach where we can actually get these numbers directly from an experimental method. But I think this is absolutely beautiful work and really, really important when we think about designing and putting together these experiments. So I should say that I'm sort of pretending that all the machine learning ideas are worked out, but they haven't been. And I think actually this particular project would test the limits of experimental design methods, and I don't want to pretend that they exist when they don't. So how to really carefully incorporate time and cost into these ideas. We have some idea about how to do that, but we need to do better. How to prioritize experiment heterogeneity and lab heterogeneity. Going deep or going broad, I think we need to really formalize this within the methods we're considering. How to have non-continuous variables. So as we've heard from the last couple of days, a lot of the parameters in these experiments are categorical. They might involve trees or lineages. They might involve count data. And these are actually less worked out than the continuous variables that we generally think about in experimental design. The scale and the size of the problem are just gigantic. The number of experimental parameters that we can consider in any one of these experiments is huge compared to what these methods are currently capable of doing. So I feel like there's some interesting ideas there. So to summarize, I think this is a really exciting opportunity for unprecedented experimental design from the statistical perspective. I really, our goals ultimately, I think, should be to maximize information gain per dollar in time, but how we design that information gain function, I think, is going to be critical to getting this right. I think this could be an interesting framework to guide coordination across members, redesign experimental proposals, and drive RFAs, but I'm actually really interested in a discussion about this because I've heard very different perspective on that from people in the audience. I also think this could really be an interesting driver for impactful experimental and methodological technologies. I think there's room to grow in both respects. So some of the discussion points I want to ask of the audience is, first of all, how would we design an objective function? I should say there's a bunch of us in this room that are already thinking about and talking about specific types of objective functions for a particular system. We're thinking about immune cells right now. I think one key point about this is there's a moving target here because as new experiments come up, as new technologies arise, as new methodologies come up, we're going to have to start thinking about how to incorporate those. I think designing this acquisition function is also going to be quite interesting, and in particular, I think explicit prioritization of diversity, we can do that with some types of repulsive functions, they're called, repulsive processes in machine learning. But how to get that right, again, I think is going to be tricky and iterative. So another discussion point that I want to bring up is, are we going to think about a general across HCA experimental design framework where we can borrow information across the different systems and help each other out? Or do we want to think about smaller system-specific experimental design frameworks? Because the objective functions, I think, truly may be system-specific. And I think also, actually, again, given the talks that we've seen recently, it seems like each system, whether it's brain or immune cells, really has its own set of experiments that are most important for understanding the basic biology of those systems. And there's also sort of a consortium culture, I think, a system culture. The final question, which I think is probably most controversial, is do we want to do this? So do we want to have some idea of a coordinated and centralized experimental design where we remove redundancy, where we maximize results per dollar, where we actually correct for biases, because there are biases in the experiments that are proposed and the systems that we think about studying. There are going to be missing areas based on maybe more rare diseases, underrepresented populations, or do we want to do it in sort of an anarchical way where we have independent, uncoordinated, and lab-driven projects. So everyone does what they do best. No one feels like they're doing anything that they don't want to do. And then we include all the work that goes above a specific bar that conforms to the policies that have been laid out in the white paper, and just hope there aren't holes. And so I'd be really interested in people's perspective on that. Thanks.